So good afternoon student. In the last lecture we have already started with active transport across the cell membrane, and in the last series we have completed about two main processes in active transport, that is primary active transport and secondary active transport. Now in this video we will deal with the bulk transport across the cell membrane. So let's start with our lecture. Under active transport in cell membrane, we have already completed number 1 primary active transport and number 2 secondary active transport. Now in this video we are dealing with bulk transport. Bulk transport is a special category under active transport in which the cell will transport the substances either inside or outside the cell in bulk amount, in large amount. So depending upon if the cell is transferring the substances either inside or outside, we are having two types of bulk transport. If the cell is transporting the substances inside the cell in bulk amount, then the process is called as endo means inside, cytosis. And if the cell is transporting the substances outside the cell, then the word for that thing is exo means outside, cytosis. So under bulk transport, if we are transporting substances inside the cell, then the process is called as endocytosis, and if we are transporting the substances outside the cell, then it is called as exocytosis. Are you clear for this thing? Now, which substances has get transported inside the cell? Then according to that, we can again classify endocytosis into two types. If we are transporting solid substances inside cell, then the process is called as phagocytosis. Okay, and if we are transporting liquid substances inside cell, then the process is called as pinocytosis. So in endocytosis there are two again types. If the cell is transporting solid substances inside the cell, then the process is called as phagocytosis, and if the cell is transporting liquid substances inside the cell, then the process is called as pinocytosis. Here phagocytosis is also called as cell eating, यानी सेल ने खाना खाया, and pinocytosis is called as cell drinking, यानी सेल ने पानी पिया. Clear? Now let's take an example of amoeba to understand this process in detail. See, the amoeba is a irregular shape of cell, and this irregular shape of cell want to transport its food, that is a solid substance, inside the cell. Now the food is a bulky molecule, it is a large size molecule, and it cannot pass through the cell membrane. The lipid bilayer will do not allow this food to pass inside the cell. So for this for taking this food inside, the cell have to follow a special procedure. Whenever this food particle will came in contact with this amoeba, the plasma membrane will do an indentation of cell membrane. It will form a fold which can accommodate the food. So here the plasma membrane will form an indentation, a fold which can accommodate the food like this. Clear? Then this furrow which is developed inside the plasma membrane will gradually deepen down and the food will shrink in that furrow. Clear? Then this furrow will further deepen down and encircle the food completely to form a vesicle attached to the plasma membrane. In this way, a plasma membrane will develop a vesicle which now having a food bulk molecule in it. This vesicle will eventually get detached from plasma membrane and it will get transported inside the cell. So that's how amoeba will transport the food particles, bulky particles inside the cell, and this whole process is called as endocytosis, as the substance is moving inside the cell, and here the plasma membrane is transporting a solid substance, a food particle, and that's why the process is called as phagocytosis. And this formed vesicle, as it contains food, so it is called as food vacuole. Clear? Similarly, a amoeba can ingest or engulf liquid substances. Okay, let's say for example these are the liquid substances which are present outside the cell, and now amoeba wants to engulf this liquid. Again it will form a indentation so that this liquid will come and shrink in this indentation. Then the indentation will further deepen down, and this watery molecule will further shrink into the indentation. Then it will form a complete vesicle which are now having the liquid inside, and this vesicle will get detached and transported inside the cell. So in this way the amoeba will transport a liquid substance inside the cell, but here the process is endocytosis. As this liquid is moving inside the cell, the process is endocytosis, but it is a liquid substance moving inside the cell, and that's why the process is called as pinocytosis. Are you clear for this thing? Now what happens inside the amoeba? This food will get digested with the help of lysosomes. So there will be so many lysosomes will come and attach to this vesicle. Here is the food, and due to the lysozymes which are present in lysosomes, the lysosome will release a lysozyme on the food material and the food will get digested. Now after digesting the food and utilizing the material from the food, the vacuole will now contain some of the undigested food material and some of the waste product of food material. Now amoeba is not in requirement of these substances, so amoeba will throw out these substances outside the cell, and that process is called as exocytosis. As the substances are now moving outside the cell in bulk amount, the process is called as exocytosis. For that process, this vacuole will eventually come and fuse with this plasma membrane. This vacuole is having undigested food material as well as waste products, and now the plasma membrane will open up like this to throw these substances outside the cell. Okay, so this process is called as exocytosis because the substances are moving outside the cell in bulk quantity. So just to summarize, in bulk transport we will transport the substance in bulk amount. हम large amount of substance को transfer करेंगे either inside the cell or outside the cell. अगर हम इनसाइड द सेल सब्सटेंस को ट्रांसपोर्ट करते हैं तो प्रोसेस को हम बोलते हैं एंडोसाइटोसिस। लेकिन कौन से सब्सटेंसेस हमने ट्रांसपोर्ट किए उसके हिसाब से, अगर हम सॉलिड सब्सटेंस को ट्रांसपोर्ट करते हैं इनसाइड द सेल देन द प्रोसेस इज कॉल्ड एज फैगोसाइटोसिस। एंड इफ वी आर ट्रांसपोर्टिंग लिक्विड सब्सटेंसेस, यानी हमने अगर लिक्विड सब्सटेंसेस को ट्रांसफर किया इनसाइड द सेल देन द प्रोसेस इज कॉल्ड एज पिनोसाइटोसिस। Phagocytosis is called as cell eating and pinocytosis is called as cell drinking. The process remains the same. कोई भी फूड पार्टिकल जब प्लाज्मा मेम्ब्रेन के कांटेक्ट में आएगा तो प्लाज्मा मेम्ब्रेन एक फोल्ड बना के उस फूड पार्टिकल को एब्सॉर्ब करने की कोशिश करेगी। फिर उस फोल्ड में फूड पार्टिकल आ जाएगा, फिर प्लाज्मा मेम्ब्रेन फिर से वो फोल्ड को इनलार्ज करेगी और करते करते पूरा वेसिकल बना लेगी और कंटेनिंग द फूड पार्टिकल्स। फिर वो वेसिकल प्लाज्मा मेम्ब्रेन से डिटैच होकर इनसाइड द सेल ट्रांसपोर्ट हो जाएगा। इसी सेम प्रकार से प्लाज्मा मेम्ब्रेन लिक्विड सब्सटेंस को भी ट्रांसपोर्ट करती है। लिक्विड जैसे ही प्लाज्मा मेम्ब्रेन के कांटेक्ट में आएगा इट विल फॉर्म अ फोल्ड एंड दिस फोल्ड विल बिकम अ वेसिकल और वेसिकल विल गेट डिटैच फ्रॉम द प्लाज्मा मेम्ब्रेन एंड विल गेट ट्रांसपोर्टेड इनसाइड द सेल। अब जो फूड वैक्यूल अंदर बन गया उसमें का जो फूड है वो अमीबा विल डाइजेस्ट एंड आफ्टर डाइजेशन सम ऑफ द फूड विल रिमेन अनडाइजेस्टेड एंड सम वेस्ट प्रोडक्ट विल आल्सो फॉर्म। नाउ ये जो वेसिकल है अब इसका अमीबा को कुछ भी काम नहीं है तो ये वेसिकल फिर से आएगा और प्लाज्मा मेम्ब्रेन के साथ फ्यूज हो जाएगा और इसके सारे कंटेंट्स वो बाहर फेंक देगा। इस प्रोसेस को हम बोलते हैं एक्सोसाइटोसिस। But for doing all this process we require a huge amount of energy and that's why this transport comes under active transport across the cell membrane. So this is all about the active transport across the cell membrane. Now in the next videos we will try to learn about the cell organelles in cytoplasms. Thank you.